My name is Ron Najafi. I'm CEO of Emery Pharma. It's a pleasure hosting Dr. Julia Minitti. Julia did her Ph.D. at Brown University, her J.D. at Santa Clara University. She spent a few years at the Albany Molecular Research in Albany, New York, prior to her Ph.D. As a bench chemist. So she is a chemist. She did chemist methodology, synthetic methodology, which is really my background. And she's been practicing patent law for the last 7, 8, 9 years at Wilson Sonsini, a very premier law firm in San Francisco Bay Area and perhaps in the United States. Julia's today she's going to be talking to us about challenges in patenting antibody drug conjugates, which is really a hot area right now. Several of our team members are actually at a conference on ADCs in Boston. We're obviously involved. Many of you are involved in antibody drug conjugate analytical methodologies and bioanalysis. So I thought it's good to learn some of the challenges in patenting ADCs. So here is Julia.
Great. Well, thank you, Ron, for having me here today. I'm going to talk a little bit about patents, getting patents to antibody drug conjugates, and hopefully this is interesting to all of you. Feel free to ask questions during the talk if you have any. Because I'm a lawyer, I have to give my disclaimer at the beginning. So my comments here do not reflect my firm. They reflect my own comments and my own opinions. So today I'm going to talk a bit about ADCs generally and how we patent them. I'm going to talk a bit about some of the challenges we're seeing to antibody patents, as well as challenges to linker patents. As we all know, antibody drug conjugates have the antibody and the linker bound to the payload. And then we're going to talk about strategies for obtaining ADC patents, and finally some other ADC considerations. Many of you probably know this already, but antibody drug conjugates are molecules that include an antibody, which is referred to often as the targeting moiety, or a portion of an antibody, as well as a covalent linkage system. Now, this could vary between a cleavable linkage system or a non-cleavable linkage system, but many of them are sort of well-established, well-known in the field. And finally, there's the payload. The payload, in many of the cases of ADCs, we're discussing cytotoxic payloads, but we might also be talking about other payloads like immune stimulatory payloads or radioisotopes. So other molecules that you want to get to the site that the antibody is drawn to. ADCs are a growing sector of approved drugs. So they're the fastest growing drugs in oncology. And as of September 2023, there's 13 ADCs have been approved by the FDA, and 7 have been approved by the EMA. So target antigens of approved ADCs include CD33, CD30, CD22, immune cells, HER2, Nectin-4, etc. The cytotoxic payloads of approved ADCs are commonly recycled. So you probably heard of MMAE or deruxtecan, which is exatecan, the topoisomerase inhibitors. In that, these are very cytotoxic agents that are targeting cancer cells and are often reused. The linkers of approved ADCs are primarily cleavable linkers, enzymatically or chemically, although I will say this area has been growing. I'm seeing a lot more innovation in the linker space over the past couple of years. Just like any other drug, patent protection around ADCs is pretty essential. The cost of developing a drug and getting it to market, as many of you know, is very high, hundreds of millions to up to $3 billion to get a drug from early stages to commercialization. We're seeing a lot of investment by VCs in ADC technologies. That's been growing since 2018. And we think there's about $1.8 billion invested in ADCs by the venture capital firms. So it's definitely an area of interest and focus. Patent protection is very important to get these investments, though. So you want to be able to show that you have exclusivity around your antibody drug conjugate in order to get those investments, grow your small company, and eventually get your product to market. I'm going to start sort of simple here before we go into the nuts and bolts of how the issues with patenting ADCs and some of the challenges we face and some of the challenges that are facing the patents that are issued in that space. I just want to talk about patentability first. So what's the requirement of a patent? So to get a patent, there are 4 main statutory requirements. There's Section 102, USC Section 102, that requires that your invention be novel. Section 103, that requires that your invention be not obvious. Section 112 is split into 2 parts. It's written description and enablement. So written description, and these can get confused a little bit, requires that you describe your invention to show that you were in possession of the invention at the time it was filed. So you need to adequately describe the full scope of your invention. Enablement's a little bit different, although they often get confused even by the courts, even by the Supreme Court. The enablement requirement says that you have to teach someone of ordinary skill in the art how to make and use the invention without undue experimentation. So as a scientist, I think it makes a little bit of sense to us when we read a paper, are we able to read that paper and reproduce the invention, right? That's enablement. Are there enough experimental procedures? Is there enough data in the paper to enable us to make and use the invention? Whereas written description is a little bit more of, if you've described a broad scope of antibody drug conjugates, have you adequately described all of the members of that group? Have you described which antibodies you could use? Have you described which payloads you could use? So that's description, not necessarily examples, it's just describing your invention. Okay.
I have a quick question.
Sure.
Is that review process, I guess the patent office?
Yeah, that's right.
Is that someone that has a scientific background, or is that from more of a legal perspective? How would they know?
Yes, yeah. So the question is, for the evaluation of a patent to determine patentability, does the person who's evaluating the patent, who's a patent examiner, have a scientific background in order to evaluate it? And yes, with differing degrees of schooling, right? So often we'll have examiners who have maybe a master's in your area of science. Sometimes there's PhDs in the patent office. I would say it's a bit of a mixture, but they are specialized. And so when you're working with, we call it Unit 1600, if you're working with a particular unit of the patent office, the people that you're sending your patent application to will know how to search it, right? Because in order to determine novelty, they need to be able to search the chemical database to determine whether the molecules are novel. So you need to know how to use SciFinder, right? You need to know how to search the chemical databases. So, or if you have a sequence, you need to be able to search the sequence. So you do have to have enough expertise in the sciences to understand the invention and to search it. I mentioned there's been challenges to antibody patents. So, you might have heard, maybe not, but there's been a good amount of case law in the past couple of years around antibody patents. And the issue is, for many years, the patent claims to antibodies were defined by the epitope or by the antigen. So the claim would read something to the effect of an anti-CD3 antibody, right? Which would, you can imagine, cover any antibody that would hit CD3, whether it's hitting the same epitope or a different epitope as your described molecules. And so those claims were traditionally the way patent filers would go after antibodies. Well, that has been largely shut down in the US. So the courts and the patent office now have said, you cannot get every antibody that hits an antigen, right? You haven't, now going back to enablement and written description, you haven't shown enough examples of sequence diversity for antibodies that hit that antigen, and you haven't described the full scope of antibodies that hit that antigen. So on neither front, either written description or enablement, have you satisfied the requirement for patentability. Probably, you may have heard of some of these cases, but I think the one that you might have heard of the most was this most recent Supreme Court case, which was Amgen Sanofi, which was a claim to a anti-PCSK9 epitope. So the idea was the the claim was drawn to any antibody that hit 1 or 2 amino acids out of 18 on PCSK9. And the court had said, that is, you're not entitled to every antibody that hits up one or more of those residues on PCSK9. You haven't shown enough antibodies that fit that description, right? So as a result, that claim was found invalid for lacking enablement. Now, I think the situation might have been a little bit different if they had disclosed, if Sanofi had disclosed more antibodies in their patent application. So if they had disclosed 100 different antibodies that had a variety of variable regions that hit PCSK9, then they might have been in a better place. But instead, they didn't disclose a sufficient number of antibodies. So their case was found to lack enablement and invalidated. Now you can imagine, so why are people claiming this? This is what you're gonna ask me. Why do you wanna claim an anti-PCSK9 or an anti-HER2 antibody rather than claiming just the CDRs or just the variable regions, right? Well, the reason is claiming just the CDRs is easy to design around. So if another company wants to enter the space and wants to design around your invention, then all they would need to do, if you're claiming the CDRs, which everyone know what I mean by CDRs and variable regions? So it's sort of the business end of the antibody, right? The residues of the antibody that interact with the antigen. If you're only claiming those particular residues, then a third party could design around that by swapping out a residue or two. It probably still behaves the same way, right? It's not going to be that different if you swap one amino acid in the CDR, and you can get around the claims. And so the claims traditionally have tried to get breadth. You want to try to get breadth and not limit yourself to the particular sequence of the antibody, the particular business end of the antibody sequence. So that's the challenge we've had in antibody patents over the years, that the courts are very active in knocking down patents. They're saying essentially you can't get breadth around your antibody. You need to limit your claim to something very narrow and defined by the sequence in either the variable region or the CDR. So the takeaway from this, I'm not going to go into the specific cases, but the takeaway is that epitope or antigen-focused claims have been consistently rejected by the Federal Circuit and the U.S. Supreme Court for lacking enablement in written description. And that carries over to ADCs too. So this is a recent decision that came out from the PTAB, which is the Patent Trial and Appeal Board. I raised this one because I think it's kind of interesting. It concerns linkers rather than the antibody itself. So I don't know, is anyone familiar with the antibody drug conjugate linkers? Some of the common ones, you can yell it out if you know any. Okay. Some of the common ones are Val-Cit, you might have heard of that, Val-Ala. So these are sort of amino acid linkers, that usually have maybe a self-immolative group that then attaches to the payload, and maybe there's some sort of stretcher unit to attach to the maleimide that attaches to the antibody. But Seagen was one of the innovators in the space of linkers. So Seagen has a number of patents to linkers. And in 2008, Daiichi Sankyo and Seagen entered into a collaboration to develop ADCs targeting cancer cells. The collaboration ended. Daiichi Sankyo produced Enhertu, which you're probably familiar with, an anti-HER2 antibody covalently linked to exatecan, which is a topoisomerase inhibitor, and that was for the treatment of breast cancer. And non-small cell lung and gastroesophageal. And your HER2 is obviously doing very well as a product. Its projected revenue for is going to be $6 billion per year by 2026. So, Enhertu hits the market. Seagen sues Daiichi for patent infringement of their patent number here, the 039 patent. We refer to it as the last 3 digits. And so this goes to court. Often these cases go to court in the Eastern District of Texas, which it did here. So the case was heard in the Eastern District of Texas, and the final judgment was ordered Daiichi to pay an 8% royalty on sales of Enhertu for infringing Seagen's patent. So they found patent infringement. They found that Daiichi Sankyo was indeed infringing Seagen's linker patent. Okay, so Daiichi then turns around and challenges the patents at the PTAB using a process known as post-grant review. So what this is, it's an interaction with the patent office where you go back to the patent office rather than the courts and you say, I see a problem with this patent. And they challenged it based on lack of enablement due to the breadth of the claim. So Seagen's patent claim is drawn to an antibody, and this is the claim of a patent. If you don't look at claims of patents at all, they're usually one very long sentence that includes all the various parts of the molecule. And this is what's called a Marcouche type claim because it has variables in the structure. So there's the structure that's over on the right-hand side, and there's certain variables that are then defined by the very long sentence. So the sentence says antibody drug conjugate having the formula, and the formula is over here on the left right-hand side, your right-hand side. And as you can see, if you're familiar with antibody drug conjugates, they are typically, the antibody is typically bound through a sulfur, a cysteine residue to a succinimide, previously a molamide, some sort of structure unit. There's a W group here, a Y group here, and a D group here. And then you can see there's a little tiny, I don't know what letter that is, I can't really tell, P, I think it's a little P. And that says how many linker drugs are bound to the antibody. That's the DAR, if you're familiar with the drug-antibody ratio. And so here the antibody is, if you look at the claim, the antibody is an antibody. That's all, it's any antibody. So the claim is drawn to any antibody there. Okay. S is sulfur, so you didn't really need to define that, but they did. W is independently an amino acid unit having the formula denoted below in the square bracket. So this W right here is this. So that's, an amino acid. The side chain here, R19, is hydrogen or benzyl. So does anyone know what amino acids those are? Just yell it out. So if you have a hydrogen here, it would be glycine. Yeah, I heard it. Okay. Yeah, and if you have a benzyl here, it would be phenylalanine. You got it. So it basically says you've got W here. You can have a repeating unit. It says W is independently an amino acid unit having the formula denoted below in the square bracket, where R19 is hydrogen or benzyl. I think it said, does it say the number of times it can repeat? Y is a spacer unit. Okay, and then Y is 0, 1, or 2. So that's this group here, which is a spacer unit. That's all they've referred to. They haven't said what a spacer unit is. It's anyone's best guess. And then P over here, that's the DAR, is 1 to 20. That's quite a range of DARs. Okay. So what I'm trying to point out here is that in some ways, this claim is very broad, right? You could have any antibody on the left-hand side. You do have to have a tetrapeptide at W, but it can be any glycine or phenylalanine in any mix and matched order. Y is a spacer unit that's undefined. And then D is a drug moiety, so any drug can be attached there. So that's the scope of the claim. And then at the end of the claim, there's some functional language. It says, wherein the drug moiety is intracellularly cleaved in a patient from the antibody of the antibody drug conjugate or an intracellular metabolite of the antibody drug conjugate. So there's this sort of limiting functional language that they haven't defined the spacer unit or what attaches the linker to the drug, but they're saying that the molecule has this property that when it's in a patient, the drug cleaves intracellularly. So is what it is. Okay. So that was the language of the claim. Speaking of enablement, when the patent office is evaluating enablement, as I mentioned, they're looking, can a person reasonably skilled in the art make and use the claimed invention without undue experimentation? So you're looking at the full scope of that claim, that one we just looked at. Can you make and use the full scope of that invention? Can you make any antibody attached to that linker? Can you make any drug attached to that linker? Do you know what spacer is? Do you know all of the necessary procedures needed to make and use that invention? And enablement turns on the analysis of what's called the Wands factors. And there are 8 factors that go into the analysis of whether you've enabled your claims. The quantity of experimentation necessary to carry out the invention, the amount of direction or guidance presented in the reference, the presence or absence of examples, the nature of the invention, the state of the prior art, the relative skill of those in the art, the predictability or unpredictability of the art, and the breadth of the claims. So clearly the breadth of the claims in some ways is quite broad. Okay, so one problem that the PTAB found with this patent was that this functional assay at the end that I mentioned requires a lot of trial and error. So if you look at that functional assay, at the functional language at the end of the claim that said that the drug was cleaved intracellularly, that's not something you could just know by looking at that. You'd have to test that, right? You'd have to iterate. You'd have to make different molecules and see if they actually fit within the scope of the claim. That could be a lot of work, right? To try and figure out which molecules fall within the scope of the claim. You'd have to run experiments to see if the drug gets cleaved intracellularly. So the PTAB was not, it didn't look on that too favorably, as they felt that that functional assay required a lot of work. As you know, undue experimentation is work, right? That would not enable the invention. Also, the expert here commented that there would be a lot of experimentation required to create any particular antibody drug conjugate. Right? You can't necessarily just look at that and say, I know how to make any drug bound to this linker and any spacer and any antibody bound together. So, and I think probably the biggest issue they had with this, and this is something I always stress to clients when I'm working with them on patent filings, is that you need more examples. You need more working examples. And what they had here was that every single working example in the patent involved an ADC with a dolastatin or auristatin derivative. So they had a limited number of drugs. They didn't have very many examples. And they didn't have a single example of a tetrapeptide that was a glycine phenylalanine tetrapeptide in their molecules. So they had no examples to show that they'd actually made a glycine phenylalanine linker. So these are all, it's kind of cautionary tales of writing applications, and it's easy with hindsight to look at someone's patent and say you should have put more in there. But moving forward, I think that's going to be the way we have to draft patent applications. We need as many examples in there as we can get, even if some of them are prophetic and you might not perform them for a bit. So, the fact that they did not have a single glycine phenylalanine linker in there was an issue. And this patent was found invalid. So that 8% royalty that Daiichi was paying to Seagen ended after this PTAB decision. Does anyone know what the tetrapeptide is that would have glycine and phenylalanine? I don't know if you're heavy into the antibody drug conjugate space. It's the GGFG linker, which a number of different clinical candidates are using now. I think there's at least one approved drug that has a GGFG linker in it now. Glycine, phenylalanine, glycine. So, okay. So now that I've gone through all the issues that we face at the courts and the patent office, I want to talk about strategy for filing on antibody drug conjugates. So antibody drug conjugates have these 3 major moving parts. You have your antibody part, you've got your linker part, you've got your payload part. And you can imagine that you could claim it a lot of different ways. And I see claims to these molecules in a lot of different formats. So one format would just be to claim the whole thing, right? You claim the antibody bound to the linker, bound to the payload, right? That's one way to do it. You could claim the bound linker. So you could just, kind of like the one we just looked at. For Seagen's case, they focused on the linker. Really, that case was drawn to the linker because they were agnostic as to the payload and the antibody, and they were really claiming the linker, right? Alternatively, you can have a bound payload. So if you come up with a new payload, you might want to claim that either as an unbound payload or a bound payload. When I say bound, I mean an open valence. So you would literally have it bound, you'd say payload, with a bond coming out from it to the remainder of the linker, right? That's the bound payload. That's also what I meant by the bound. linker. And then there's the bound antibody. So you can imagine, just like anything else, you claim the CDRs or the variable regions of the antibody, and you say that this antibody is bound through cysteines to 1 to 6 linker payloads and be completely agnostic as to the linker payload. And then you can imagine some combination of the three where you focus on, if you focus on the sequences together with the linker, but you're agnostic as to the payload. And I'll give an example in a minute. Maybe that'll help see what I'm talking about. In addition to those, I was just talking about the bound, but there's also the unbound. So if you have a new linker and you want to claim the linker intermediate, that's the unbound form. So you'd claim the maleimide with an unbound side to it, or maybe it's unbound on the other side, depending on your synthetic procedure. But you'd claim the intermediate before it gets conjugated to the antibody or before it gets reacted with the drug. Then there's also the unbound antibody intermediate that's often claimed. So if you come up with a new antibody, we often see the antibodies claimed first, and then maybe later you would innovate around an antibody drug conjugate. So you might see a first patent application drawn to the antibody, and then a later case drawn to the ADC. And then finally, the unbound payload intermediate, which is exatecan. So claiming a cytotoxic agent by itself that would be the synthetic intermediate, right? It can't really be used by itself as a drug, but it would be an intermediate that would be conjugated to the linker at some point or reacted with to form a bond with the linker at some point. So I think the best way to think through this is to look at trastuzumab deruxtecan in HER2. So in HER2 is drawn out here where you've got the antibody. So here's the antibody. The variable regions are in this portion up here, right? That's the active portion of the molecule, the portion of the antibody that will bind to the antigen. And then these little cysteines here hanging off are the ones that are reacting with your linker. Payload here, this is exatecan, this is the GGFG linker, glycine, glycine, phenylalanine, glycine, and then here you see the succinimide is the point of attachment to the cysteine. That's the molecule, and the reason I show you this molecule, this is the approved drug, is so that I can break down the claims that could be filed on the molecule. So the full ADC, the first one I mentioned, would be trastuzumab deruxtecan. So the whole thing, what you just saw on the previous slide, the sequence of the antibody, the sequence of the link, or the structure of the linker, and then the structure of the payload. It would all be drawn out. And that's sort of like a picture claim, we might call it. That's the type of claim that you would use in court when you litigate. That's the narrowest scope claim you can get. It's very focused in on the product itself. But you'd also want to claim the bound linker. So similar to Seagen's patent, you would claim something like succinimidal pentyl GGFG, and then maybe a self-immolative group or whatever group you had that attached to the drug. That would be the bound linker approach. Then there's the bound payload approach, where you would claim exatecan bound to a linker. The bound antibody for this molecule would be trastuzumab bound to the rest of the molecule. And then you can imagine a combination of the above, deruxtecan bound to the antibody, or the payload bound to the linker, however you break it down. But part of what I'm trying to show here is that there's very many ways to claim these. And there's many ways to claim the intermediates. Yes, go ahead.
When you're claiming the full ADC patent, what is the reason claiming unbound?
What was that?
What is the reason of claiming the unbound, like a linker with the antibody or linker with the payload? What is the reason behind that?
Yeah, for sure. So the question was, what's the reason of claiming the unbound linker intermediate, or the unbound antibody intermediate? And the reason is it's a synthetic intermediate. So if someone wants to make your ADC, they're likely going to have to go through the antibody unbound, or they're likely going to have to go after the linker with the maleimide, right? Because that's the bioconjugation step.
But when you're claiming full ADC, you will report the synthesis, right?
That's right.
Not covering that one then?
When you claim the
full ADC.
That's true. They could be in the same patent application. So it's possible in one patent application, you're not only claiming the ADC, but you're also claiming the synthetic intermediates, right? Or there's a possibility that you have the synthetic intermediates claimed in a later case if you had a different synthesis for making it. Oftentimes, things could change during the course of development. As you're developing it, you might come up with a better way of creating the linker, and then you can claim that linker intermediate. Does that make sense? I didn't convince him. I think the one thing just to think about is you want to go at it at a number of different angles. So you do have your ADC case, that's great. That claim may expire. At some point we have a 20-year term for patents. I think we all know that 20 years from filing is the term of patents. And so we need to think about ways to cover the molecule maybe in different ways. So perhaps we're covering synthetic intermediates, or we're covering formulations or other aspects of the ADC. Does that make sense? And I think the other reason I raise it is sometimes I see in patent applications, people are a little fast and loose about how they write these claims. And so you'll see a claim to the intermediate first, a claim to the unbound linker, and then a dependent claim that says, and it's bound to an antibody. Well, that's a chemical reaction that changes the molecule, right? If you have a maleimide and you react it with a cysteine, that's a different molecule, it becomes a succinimide. And so that's not really dependent, it's a different molecule, right? I don't know if that makes sense, but yeah.
No. When the maleimide react with the cysteine, is going to make the
succinimide.
Succinimide is the same compound.
It's the chemical structure is different, right? The olefin's gone, right? Maleimides have the olefin, you no longer have the olefin on a succinimide. So it's a different molecule. So it's not properly dependent on the maleimide claim, right? So just thinking through how you write it, I think the claims, because there's so many moving parts, I've seen patent applications that sort of forget that there's chemistry involved there, right? It's not the same molecule anymore. You have to transform it. You're pulling a proton off the cysteine, and you're reacting the maleimide. So it's not accurate to say you just put an antibody on and it's the same molecule, right? So. Okay, and so these are many of the different ways. And part of the reason another reason I raise this is because there are so many ways to claim antibody drug conjugates, you also need to think about that when you're doing what's called freedom to operate. So when you're evaluating the landscape to see if your antibody drug conjugate has freedom to operate, you need to be looking at all of these different claim types. There might be an unbound linker, there might be a bound linker, there might be an antibody unbound, there might be the bound antibody. So there's a lot of different angles you need to come at when you're running searches to see if there's a third-party patent out there that can block your ADC from commercializing.
Potentially, they could have a claim on the linker only.
Right.
The linker only claim that prevents you from using that linker.
That's right. That's right. Yep. So we're always searching everything. So if we need to present a patent picture, a portfolio picture to an investor, we need to know, are there any blocks out there for either the linker, the payload, the antibody, the general class of antibody conjugates, right? Are there other antibody, has someone claimed a broader scope of antibody conjugates drawn to a particular antigen? Well, those are hard to get, but there are some patents out there that have snuck through the system. And different jurisdictions allow different patent scopes. So for example, the US is very tough right now on enablement and written description, as I've mentioned. Europe has a different toughness. So Europe is also a bit tough on getting antibody patents, but they will allow broader scope claims if you can show a benefit of your antibody that hits an antigen relative to a known antibody. So if you can say if you have better binding or better affinity for that a target or a different epitope that gives some improved properties, then you're going to get a broader claim, and you often can get a broader claim in Europe. So, okay, so what about the DAR? The DAR is really important too. So I have some companies that are innovating around DAR. They, the idea of a controlled DAR, being able to have consistent DAR throughout their ADCs, or very low DAR, very high DAR, right? Kind of go every different direction. A lot of times the cytotoxic payload is a little greasy, so you want to offset the greasiness of the payload with the polarity of the linker. And so you've probably seen some advances in increasing the polarity of the linker to make the antibody behave more like an antibody and less like a greasy molecule. So that could be a big part of your innovation. You could hinge your claim on your DAR if that's something important to your innovation. Being able to tune your desired DAR is important, and you need to include examples of how you achieve your different DARs. So if your claim, your innovation is that you're able to achieve consistently a DAR of 2, then you better show in your application how you consistently achieve your DAR of 2. So seeking enablement and written description, sort of the theme here is you need examples. You need as many examples you can come up with. And I think in this sort of, AI, ML, computational world now, I'm seeing a lot more capability from companies to generate, even computationally, molecules that could bind to an antigen or molecules that could adequately describe the full scope of the invention, even if you haven't tested every single one. And so to the extent you can, you really want to get the examples that you've run, but maybe some prophetic examples as well too, that I haven't run this, but I've run some modeling, I've run some different ways of looking at the antigen, and I think that this type of CDR, a variable region, would be active. And here's sort of my scope based on molecules that I've made and molecules that I could make. You want to show, if you want to get a broader scope claim, you want to show a variety of payloads. So for that CGen patent, perhaps if they'd shown 5 or 6 different payloads attached to the linker, they would have been in a better place. And they also probably should have shown the actual linker that they claimed in their examples. So, if they had shown GGFG linker and maybe an FFGF and a GFGG and a variety of different linkers there, they would have probably been in a better place when it came time to assert that patent. What else? Show that you're able to bioconjugate to different antibodies. So different payloads, also different antibodies. You need to be able to show a little bit of versatility. If you're only bioconjugating it to one antibody, then you're probably going to be narrowed by the patent office to either that one antibody or one class of antibodies. So try to show diversity on all aspects. And then the scope of your examples will ultimately determine the scope of your claims. I think if you get nothing out of this talk, examples are critical to the scope of your claims. If you have one linker described and one payload described, then you probably are not going to get every linker and every payload allowed by the patent offices. And then consider prophetic examples if you don't have experiments done. Okay, other ADC considerations, timing. So whenever we're working with a client, we're always thinking about what did they make first? What do they have data for? We need to support the antibodies and the conjugates with data. We need to, if you're familiar with patent prosecution, you start usually with a provisional application and you have 1 year to add data. So you need to time it out and see, are we going to have data in 1 year? If we're not going to have data in 1 year, then we probably want to hold off on filing the ADCs. But we do have data on the naked antibody, so let's file on that one first. So you can think about the strategy based on when you're going to have data. Then I mentioned freedom to operate. So that slide that I showed before with all of the various ways that you could potentially claim an ADC, you need to be able to think about searching the landscape for third-party patents that could block your ADC from launching. And then, as I mentioned, patentability for antibodies is different throughout the world. So to make it even more complex, every country has its own standard. So where you might have the perfect solution, this claim is the perfect claim for the US. Well, you can't stop there. You also have to think about what's a good claim scope for Europe, and that needs to be in the patent application at the time of filing. You can't just make that up later. You need to have the claims in the application. You need to have an attorney who's prosecuted throughout the world so that they can say, well, that's going to work out well in China, but that's not going to work out well in Europe. So you need to be drafting your claims appropriately. And then I guess finally, you should work with your patent attorney on both patentability and FTO, because for ADCs, it's complex. It can be a complex question. There's a lot of moving parts. So I think that's it. And happy to take any questions.
Julia, what about...
Yeah, thank you.
Julia, what about this freedom to operate, where you find something, you find another patent that says, no, I can't, essentially, I don't have permission to operate because they have a patent. But when you examine the patent, you see it's too broad, too ridiculous of a patent, and you say, how can the examiner give this patent to them?
Right.
They don't have example, they don't have novelty, they don't have, there's lots of anticipation in there. Do you have any mechanism to essentially go to PTAB prior to essentially filing your patent?
Well, it shouldn't affect you filing your patent. Your ability to file your patent is different. If you have an invention, you should file your patent. Freedom to operate is another story. So freedom to operate is just, is there someone else who's going to block you? And if there is, you need to come up with a position on it. And yes, PTAB would be a place you could go. You could challenge the patent at the patent office. That's essentially what Daiichi did to Seagen. They said, listen, we just got sued on this, but the patent examiner gave them way too much, so we're going to challenge this. We're going to go to the patent office and we're going to say this is too much. They got too much. And so there is that mechanism that you could challenge it. I think a lot of times for early stage companies, they're not Daiichi, and so they don't necessarily have the same resources to litigate. Even sometimes PTAB proceedings or post-grant proceedings can be quite expensive. So they might at least come up with just a patent opinion or some opinion of counsel that, we don't think you should worry about this patent for these reasons. And it's a well thought out explanation of why the patent's not valid or why the client's not infringing it. Yeah, that's a good question.
Do you ever run into issues with obviousness or novelty when using a commercially available antibody or a commercially available payload?
Absolutely. Yeah. So the question was, do you run into issues of novelty or obviousness when you're using commercially available components? And yes, if you were, you can't be novel if you're using someone else's. So for example, if I'm using exatecan, that alone is not going to get me a patent because someone else invented that. They've already disclosed it. I need to have something attached to that exatecan that's going to make it novel. And maybe it's the antibody or maybe it's the linker, but I have to be novel, right? Now, and for inventiveness, that's always a little bit grayer. So inventiveness is, let's say I invented a new linker, it's GGFG, but I swapped out a phenylalanine for an arginine or something. Is that obvious? Well, maybe. But the way you would overcome that rejection is you'd show some unexpected benefit. So if I swapped out one of the glycines for an arginine, all of a sudden the molecule behaves incredibly differently, and it's got a different profile in the body. And so that is inventive, right? No one would have thought that would work so well. So that's how you'd overcome an obviousness rejection. Yeah.
How much unexpected result do you need to show?
It depends how close it is to the art, I would say. So the question was, how much unexpected results do you need to show to overcome an obviousness rejection? And it really depends on how close you are. So if your modification, let's say your linker is only modifying, instead of a GGFG, it's GF-GF, that's pretty similar. And the examiner is going to run their search of linkers. They're going to come up with the GGFG linker that's worked quite well with a lot of different antibodies and payloads. And they're going to say, well, that's pretty obvious. You just kind of moved the amino acids around. You didn't really do anything. And so if it's really close, you need more data. If it's further away, you likely need less, if not none. So it's all a question of sort of distancing yourself from the closest prior art. Yeah. And also the prior art is not looked at. When the examiner looks at the prior art, they don't look at, let's say there's 400 linkers out there. They're going to look at the better ones. They're not going to look at the ones that are almost never tested or used. They're going to look at the ones that are appealing, right? Because one of skill in the art would start with the better ones. They would start with the ones that have been used, tested, tried, have been shown to be, tolerated, are cleavable. So those are the ones that you want to be furthest from, the ones that are, I guess, desirable in the space. Yeah.
So what happens, suppose the small change in the linker improve your therapeutic index as well as minimize the off-target toxicity? Like, suppose example is a phenylalanine in the tetrapeptide.
Uh-huh.
If somebody just put the small functionality like fluoro or methoxy, and
right.
got the very good activity, not activity like drops, drops of oil or this kind of thing. What happened? It's come under selection patent, or what, how does it go there?
So yes, it could be if your original filing, sort of generically covered that, but it didn't cover the fluorinated or the methoxy form of it, then yes, you could file a later filing and say you'd show patentability to the patent office. You'd say this is patentable because no one would have expected that adding a fluorine would have changed the tox profile, right? It's such a small portion of the full antibody drug conjugate, like if adding one little thing. So that's the way you would respond to the patent office when you got that sort of rejection. Now, I will say in this, because I'm more of a small molecule person by training, I always advise people to go a little further than a fluorine, just because it's isosteric to a hydrogen. It's very similar. So unless there's some unexpected improvement outside of sort of solubility, maybe that's usually not enough to get you patentability, barring spectacular unexpected results, right? But
when you improve on someone else's patent and you get unexpected results, you still need to use this other company's patent to get your unexpected result. Do you need to get the license from that company to practice?
It depends. So first thing we look at is patent term. So let's say the patent of concern expires in 2028 and you're not going to launch your product until 2032. Then no, you probably don't, right? You're covered under the safe harbor. You don't really need to do anything with that. Let's say the patent that is of concern expires in 2035 and you plan to launch in 2032. Then you either need to think about a non-infringement position, maybe you design around that patent, or you maybe think about a license.